So next we're going to hear from Javier Blanco. Javier is a full professor in pharmaceutical sciences at the University of Buffalo. He's had 18 years of successful independent research and funding in translational oncology, pharmacogenetics, cardio-oncology, and drug metabolism and the disposition of Down syndrome. So Javier, welcome, and we're so glad to have you here.
Thank you, Greg, for the introduction. We'll start sharing my screen. I hope you can see it. Okay, so what I'm showing here is perhaps one of the oldest reports that I was able to find describing a case of Adriamycin cardiotoxicity in a child with Wilms tumor. And this is to highlight the fact that we're dealing with an old problem, but it's still very relevant. I will argue that we have a very good understanding of the mechanisms that mediate anthracycline-related cardiotoxicity, but still we're trying to define which covariates are important in terms of modifying the risk for individual patients. This is a typical chemotherapeutic protocol for acute lymphoblastic leukemia in children. And as you can see, it's a rather complex protocol that involves the administration of various drugs, including high-dose methotrexate, of course, anthracycline, 6-mercaptopurine, dexamethasone over a period of up to 2.5 years or so. What I'm trying to highlight here is that this represents quite a significant clinical challenge in terms of balancing efficacy and toxicity in these patients. As Dr. Bhatia mentioned, her group pinpointed a very significant and clear dose-response relationship between the odds ratios for developing cardiomyopathy and total cumulative exposure to anthracyclines. As you can see in the right-hand side of the plot, there is an almost exponential increase in the risk for cardiomyopathy in patients that have been exposed to doses higher than 200 milligrams per meter square, which are not uncommon doses in pediatric oncology in general. And again, referring to Smita's talk, it is known that certain factors like concomitant radiation to the heart, female gender, younger age are modifiers for the risk of cardiomyopathy. And the question that we have been trying to answer is whether there's a role for genetic susceptibility or genetic factors that will modify risk. What I'm showing here is a very simplistic depiction of what we believe is going on in terms of the main mechanisms that mediate cardiomyopathy. This is a cardiomyocyte. And again, these are not all the pathways. It's just an oversimplification. And when I would like to focus your attention to this area of the figure in which I'm showing an enzymatic pathway that mediates the conversion of doxorubicin, the parent drug, inside the cardiomyocyte into these alcohol metabolites, doxorubicinol or downorobicinol for downorobisin. And now we know that this simple catalytic reaction is catalyzed by at least 4 or 5 myocardial carbonyl reductases. This reaction we believe is important because alcohol metabolites derived from the parent drug are more cardiotoxic. They, for example, are 40 times more potent in terms of mediating perturbations in iron metabolism inside the cardiomyocyte, calcium handling, and they do damage mitochondria inside the cell by direct and indirect mechanisms. So our thesis has been that perhaps if we understand variability in this simple enzymatic step mediated by carbonyl reductases, we may contribute to the understanding of risk for cardiomyopathy. By the way, I'm just showing you the fact that carbonyl reductases catalyze the reduction of this carbonyl group right here. This is carbonyl 19 in the chemical structure of a typical anthracycline into this alcohol metabolite. So when we started to work on this topic 17 years ago or so, there was already evidence from murine studies. This is probably one of the rare examples in which murine studies were able to contribute some information in terms of risk factors for cardiomyopathy that indicated that overexpression of the human enzyme in the heart of mice accelerated anthracycline cardiomyopathy. And the opposite was also true, meaning that knockdown one CBR1 allele, one of the carbonyl reductases, protected those animals from anthracycline-related cardiomyopathy. So with that in mind, we decided to go ahead and study these genes. We did studies in gene regulation, gene expression. As Smita mentioned, we humans have two carbonyl reductases, CBR1 and CBR3. Both genes are located in chromosome 21. And this is showing that the genes are expressed in various tissues. Of course, they are expressed in heart, liver, brain, and other tissues. This is just to show the chromosomal location of both genes. They are close together. The theory is that CBR3 has been, is a gene that originated later on during evolution because of a gene duplication of CBR1. As I said, they are located in chromosome 21. Relatively simple genes. As you probably know, individuals with true copies of chromosome 21 have Down syndrome. And it is known that pediatric patients with Down syndrome are at very high risk for developing childhood acute myeloid leukemia and childhood acute lymphoblastic leukemia. So this is an example of a therapeutic protocol by the Children's Oncology Group. This is a paper from 2008 that documented a rather unusual incidence of anthracycline-related cardiomyopathy in pediatric patients with Down syndrome and acute myeloid leukemia that were treated under POG 9421. The incidence was very high. 17.5% of the patients developed cardiomyopathy. So we review the literature on Down syndrome and acute myeloid leukemia, and drawing universal conclusions is difficult because the literature is scarce. There are differences between chemotherapeutic protocols, differences between the types of anthracyclines, differences between how clinicians define cardiomyopathy. But the overall conclusion that we made is that patients with Down syndrome can achieve favorable therapeutic outcomes after receiving treatment with reduced doses of anthracyclines. In 2010, we did a pilot study examining the expression of CBR1, one of those genes located in chromosome 21. It's just a small collection of heart tissue samples from donors with Down syndrome and non-Down syndrome. This is myocardial expression of the protein, just 4 heart tissue samples for the Down syndrome group. You can see there is a trend towards higher CBR1 expression when, as determined by a technique known as Western blot. There was a trend.
I'm going to interrupt just for a second. There's a box over the lower part of your slides that we can't see the data. Can you move or reduce that box?
Yeah, am I moving it? Let's see. Can you? That is not better, right? Now it's at the top.
We still see it in the lower portion. That's okay. If you can't move it, just continue on and describe it. We just can't see that little lower portion of the slide. So
no
problem.
I don't know what's going on because I can see it in my screen. Sorry.
Okay, no worries.
Well, there is a trend towards increased CBR1 activity, enzymatic activity. Can you see the slide? Okay, is the slide okay now, Greg?
No, it still has a rectangle over the lower portion, but that's okay. You keep going, and we can see the vast majority of the slide.
I don't know what's going on. Okay. All right, I'll keep going.
Yes, no worries.
I apologize for that. So we then expanded these studies by including more heart tissue samples. This is just to show an independent collection of samples from donors with and without Down syndrome. Again, we were able to document increased CBR1 expression at the mRNA level, left-hand side, protein level, right-hand side, in heart tissue from donors with Down syndrome, which is in agreement with the expected gene dosage effect for these samples. Now we know that this relatively simple enzymatic reaction is catalyzed by more than one of these carbonyl reductases. And this is true for any type of drug metabolizing step that you can think of. There is always enzymatic overlap. So we compare, for example, the ability of those heart tissue samples to synthesize downrubicinol, the cardiotoxic metabolite, in samples with and without Down syndrome. And as you can see right here at the top, I hope you can see the slide. enzymatic activity, the rates of enzymatic activity were similar. However, our data suggests that for samples from donors with Down syndrome, CBR1 appears to be the main driver. You can see that the regression of CBR1 protein content in myocardium versus enzymatic activity is tighter for the Down syndrome samples than for the non-Down syndrome samples. And this is a relatively simple linear regression model that suggests that incorporation of CBR1 protein content and other aldoketo reductases, these are other anthracycline metabolizing enzymes, improves our ability to predict the synthesis of cardiotoxic doxorubicin in myocardium. This is just to show that the variability, again, my apologies. This is to show that the expression of these drug metabolizing enzymes in Down syndrome and non-Down syndrome myocardium, as you can see, is quite variable. So we're trying to develop better models that will improve our ability to actually predict the synthesis of alcohol metabolites in myocardial tissue. And this is a more recent study in which we use classification and regression tree analysis. I'm not going to discuss all of the data, but as you can see at the top, there are many or different covariates, including gender, Down syndrome status, and of course the abundance of these reductases in myocardial tissue that helps to improve whether these heart tissue samples will exhibit low, medium, or high catalytic activity. So we like to believe that these findings have contributed a little bit of information to support the rationale for those reduction strategies in more recent protocols by the Children's Oncology Group. This paper was published in 2017, describing this new protocol that incorporates a 25% reduction in the total cumulative dose of daunorubicin for these patients. Now I'm going to move forward and describe our studies with Smita Bhatia and my colleagues from the Children's Oncology Group. And again, I will refer to Smita's presentation by saying that we characterize a variant in carbonate reductase 3. This is another one of those anthracycline metabolizing enzymes that is expressed in human myocardium. The variant is quite common in individuals from European and African ancestry. And this is an oversimplified cartoon showing that the valine isoform is twice more active, catalytically active than the methionine isoform. Again, we perform quite elegant pharmacokinetic studies, enzyme kinetic studies to characterize these two isoforms. And on the right-hand side, I'm showing you a look at the catalytic site of both enzymes. This is a study that we did in collaboration with Deb Ghosh from the Hotmont Woodward Structural Biology Institute. And basically the conclusion is that the faster isoform of CBR3, the valine isoform, is more efficient in terms of accommodating the NADPH cofactor in the active site than the methionine isoform. So this is the explanation for what is going on in terms of differences in catalytic activity. As Smita mentioned, her group recruited 165 cases with documented cardiomyopathy and 323 controls matched for a series of variables. This is to show that cases were treated for different types of primary cancers, acute lymphoblastic leukemia, bone sarcomas, soft tissue sarcomas, Hodgkin's lymphoma. We had echocardiographic findings for the majority of cases. And the main figure for that study is this one that shows that individuals with 2 copies of this fast isoform of CBR3 were at a higher risk for developing cardiomyopathy than individuals with 1 or 2 copies of the variant allele that dictates the slow isoform of CBR3. Is everything okay, Greg?
Everything looks great.
All right. So we recently expanded these findings by conducting a single-arm observational pharmacogenetic study here at Roswell Park in Buffalo, New York. We were able to enroll 155 patients, but 92 of those patients, we had complete echocardiograms at baseline ×0 and at 6 months after the initiation of chemotherapy with doxorubicin. And again, these are adult patients with breast cancer. But what we saw was quite intriguing and interesting. At 6 months, there was a significant decrease in left ventricular ejection fraction for individuals with 2 copies of the fast isoform of CBR3 in comparison to individuals with 1 or 2 copies of the slow isoform of CBR3. The decrease, the magnitude is around 7.5%. We can argue whether that's relevant or not. That's an issue that cardiologists can discuss, but this probably represents a snapshot of what's going on in those hearts at an early time point, 6 months. So I'm going to briefly talk about the genome-wide association study, again, that was performed by Smita Bhatia. She was the study leader, and her group. She was one more time able to recruit 162 cases of documented cardiomyopathy, childhood cancer, of course, I'm referring to childhood cancer survivors, and 262 controls. This was a genome-wide association study, and the difference with the previous study is that at this time, instead of looking at 2, 3, 4, 5 single nucleotide polymorphic variants, in this study, Smita was able to interrogate over 500,000 single nucleotide polymorphic variants. So it's a tremendous improvement in terms of the extent of coverage for genetic variants. As Dr. Bhatia mentioned, they identify a significant association coming out from the SELF4 gene, association with the risk of cardiomyopathy. And again, I'm referring to Smita's finding. They demonstrated a very nice dose interaction between having 2 copies of the G allele in SELF4 and the risk of cardiomyopathy, especially the interaction was quite significant at high doses of anthracyclines, higher than 250 milligrams of total cumulative exposure. So what do we know about SELF4? SELF4 is one of the members of the SELF family of mediators of splicing. Self family members mediate the splicing of many important genes, including cardiac troponin. As many of you probably know, cardiac troponin has an essential role, a structural and functional role in human myocardium, and for example, participates in calcium signaling. So during embryonic development in the human myocardium, the form of troponin that predominates are these embryo variants of troponin, TNNT2 embryo forms. In adulthood, there is a predominance of expression of the adult isoforms of troponin. It is now known that individuals with cardiac disease tend to present a relatively high proportion of embryonic isoforms in troponin in the myocardium. And this perhaps correlates with changes in calcium sensitivity and the development of cardiomyopathy. So we decided to examine whether the genotype in SELF4, the genotypic variant that was significantly associated with the risk of cardiomyopathy, somehow was related to the relative content of troponin isoforms in myocardial tissue. And this is what we saw, is that having 2 copies of the G allele of the SELF4 variant linked to the risk of cardiomyopathy in general correlates with the expression of both the adult isoforms of troponin and the embryonic isoforms of troponin in myocardial tissue. What I'm showing here is our agarose gels showing the bands for troponin isoforms, but I should say that we actually quantitated the expression by using real-time polymerase chain reaction. We also examined the expression of these troponin isoforms in myocardial tissue samples from donors with and without Down syndrome. So here in the left-hand side is the expression of fetal troponin isoforms, the adult troponin isoforms, 3 in the middle and 4 on the right-hand panel are also shown. as you can see, there was a trend towards increased expression of these fetal troponin isoforms in the myocardium from individuals with Down syndrome. And we are trying to elucidate if that correlates with the risk for cardiomyopathy. I should say that the expression of fetal troponin isoforms in myocardium from adults in general is quite low. It represents 1% or lower, right, than the adult isoforms of troponin. And I'm quickly show you some of the experiments that we're doing. We're using human-induced pluripotent stem cell-derived cardiomyocytes. I am pretty sure that Dr. Burridge will talk in more detail about this model for the study of cardiotoxicity. And let me tell you just briefly what we're trying to do in the lab. It turns out that there is a gene known as DIRC1A that also participates in the splicing of troponin in adult myocardium. And DIRC1A is a very prominent gene in the pathobiology of Down syndrome. DIRC1A is also encoded in chromosome 21, and individuals with Down syndrome have increased expression of DIRC1A. So if one overexpresses the DIRC1A protein in these cardiomyocytes and measures the relative abundance of troponin isoforms in those cells, what we can see is a shift towards an increase in the relative abundance of these fetal isoforms in the cardiomyocytes that resembles what we saw in the myocardial tissue samples, okay? If we are treating those cells with daunorubicin, for example, and measuring the flow of calcium at the intracellular level, and I'm going to quickly show you what we are seeing. So after treating the cells with daunorubicin, you can see that these are measurements of calcium flow that look like electrocardiograms, right? This is fluorescence. And you can see that the addition of daunorubicin for just 4 hours disturbs the flow of calcium in comparison to the controls right here at the top. In cells that overexpress DIRC1A, under basal conditions, the flow of calcium appears to be similar to controls, right? But after treatment with daunorubicin, you can see an effect. The pace, there are perturbations in the rhythm, and also, but they are not as marked as in the cells that were not expressing DIRC1A. So it seems that the overexpression of DIRC1A is somehow protecting, quote unquote, these cells from the toxic effects of daunorubicin. And I'm going to just close by showing you these pictures, or rather movies, that show the flow of calcium. So these cells have been stained with a dye that is sensitive to calcium. So when calcium binds to the dye, there is fluorescence. And you can see on the right-hand side that the cells have a more, quote unquote, normal beating pattern than the cells in the right-hand side panel. I think I said it right? So these are really cool experiments. This is like watching cardiotoxicity in action, right? You can see the effects. Normal on the left-hand side and some perturbations in rhythm in the right-hand side. So that's all what I have to say. I would like to thank the members of my lab that have done all this amazing work. And I will particularly thank Dr. Smita Bhatia for allowing me to participate in those great studies from the Children's Oncology Group. And finally, I would thank my colleagues, my wonderful colleagues from UB, University of Buffalo, and Roswell Park, Dr. Tracy O'Connor, Dr. Umesh Sharma, Dr. Rachel Hageman-Blair, and Dr. Jennifer Lang. And I'm also very grateful for the financial support from the National Institutes of Health. Thank you.
Thank you, Javier. That was a wonderful lecture, and thank you for persisting through the technical difficulties. We apologize for those, but that was great. I'd like to remind everyone that we will be taking questions, so please put them in the Q&A box. We'll be tackling those questions at the end of the session.